Hello, my dear student. This is the second lecture in cell biology with following topics. So the topics are for today sizes of the cells, the nature of biological molecules, and also we must discuss macromolecules today, such as carbohydrates, lipids, and nucleic acids. Let's start with the sizes of the cells and their components. What measurement we are using in order to describe some parts of the cell. The two common measurements are used, like micrometer and nanometer. For example, I want to remind you that 1 micrometer is equal to 10 to minus 6 meters, and 1 nanometer is equal to 10 to minus 9 meters. But also we can use angstrom as an unit, additional measure, which is equal to 1/10 of a nanometer or 10 to minus 10 meters. But please take into account that angstrom is commonly used by molecular biologists for atomic dimensions. So let's describe some examples. For example, highly elongated proteins such as collagen or myosin are over 100 nanometers in length. Yes. For example, if we take DNA molecule, it's approximately 2 nanometers in width. And complex molecules such as ribosomes, microtubules are between 5 to 25 nanometers in diameter. But still, this is quite smaller molecules in our cells that can be seen only with the help of electron microscope, because they are quite small organic substances, and we can't see, for example, ribosomes with the help of standard light microscope. As for other bigger components of the cell, like organelles, for example, and other components, for example, nuclei are approximately 5 to 10 micrometers and not nanometers. Mitochondria are approximately 2 micrometers in length. Also, prokaryotic cells typically range in length from about 1 to 5 micrometers, and eukaryotic cells from about 10 to 30 micrometers in length. So these parts we can usually observe under standard light microscope, and you see here mitochondrial, bacterial cell, chloroplast, also lymphocyte as a blood cell, and epithelial cell as a type of eukaryotic cell. So all these components we can see using standard light microscope. But for example, frog egg can be seen with the naked eye because it's approximately 2.5 millimeters in diameter and not micrometer. So we can see it clearly by our naked eye. So now about the second part of our lecture. This is chemistry of life. And I want to say that it's impossible to begin understanding cellular function without a reasonable knowledge of the structure and properties of the major types of biological molecules. So let's describe which type of organic substances can be observed within the cell. These are 4 different types of organic substances, like macromolecules, building blocks of these macromolecules, also metabolic intermediates, so-called substances that are derived during cell metabolism, and also molecules of miscellaneous function, like vitamins, ATPs, or some steroids. Today we are going to discuss macromolecules and building blocks of macromolecules. Please remember that in most type of cells we can meet 4 different types of macromolecules, 4 different classes of organic substances. These are proteins, nucleic acids, carbohydrates, and lipids. Please take into account that first three types, proteins, nucleic acids, and carbohydrates, are polymers composed of a large number of low molecular weight building blocks or monomers, and with the help of process that we call polymerization or polymerization reaction, these monomers are attaching to each other to build up finally um, polymers as macromolecules. And you see some example of it. For example, these building blocks are monomers that are constantly attaching to each other by the reaction of condensation, which means release of water molecules. And these monomers are like a building blocks that usually build up the three-dimensional structures of polymers. This is very good picture depicting the distribution of these organic substances within different parts of cells. For example, you see cell wall with carbohydrates, especially cellulose, yes, as a main component of cell wall. Also, starch belongs to carbohydrates, for example, and you see starch grains in chloroplasts. You see structure of plasma membrane, for example, that is constructed with the help of lipid molecules. Phospholipids are here, and also proteins, especially we see here integral protein as a part of a plasma membrane, and also carbohydrate component of the plasma membrane. And these carbohydrates or very often are attached to proteins or attached to lipid parts of the plasma membrane. So now very important is to remember and to learn what are the building blocks of macromolecules. I mean these four classes of macromolecules. Yes, sugars are building blocks for polysaccharides, for carbohydrates. Amino acids are precursors or building blocks for proteins, of course. Nucleotides are precursors or building blocks for nucleic acids. And as for lipids, for example, we can say that fatty acids are essential part of lipids, but also in case of lipids, we have some another groups like glycerols. Yes. Let's start describing each class of these organic substances, these macromolecules. First is carbohydrates. It includes, of course, simple sugars, so-called monosaccharides. It includes disaccharides and also polysaccharides. Yes. The main function of carbohydrates, they function primarily as source of chemical energy and also as durable building material for different kind of biological construction, like cell wall, for example. We already know about it, that cellulose is the main component of cell wall. Sugars containing 3 carbons are known as trioses, with 4 carbons as tetroses. Those with 5 carbon atoms in their molecule known as pentoses. But carbohydrates, monosaccharides that contain 6 carbons are known as hexoses, for example. And we can say that, for example, glucose molecule belongs to hexoses because it contains 6 carbon atoms. And for example, ribose as a part of nucleotide belongs to pentose. Please remember these monosaccharides as the most common monosaccharides that we meet. These are glucose, of course, fructose, galactose, and also 2 pentoses like deoxyribose and ribose that we have in molecules of DNA and RNA. Please remember also here that deoxyribose is a pentose that builds up DNA nucleotides, and ribose participates in case of RNA nucleotides. Yes. Disaccharides are also carbohydrates that contain two monosaccharides, and these two monosaccharides are covalently joined with the help of glycosidic bonds. You see here formation of glycosidic bond, for example, between glucose and fructose, and eventually we have the saccharide sucrose, yes, as a product. Also, the another representative of this saccharide, also very popular representative, is lactose. But we have some different composition because instead of fructose, lactose contains galactose, another monosaccharide. So in case of lactose, we have glucose molecule and also galactose molecule, yes, joined by the same glycosidic covalent bonds. As for polysaccharides, we must remember at least three representatives of polysaccharide, like glycogen, starch, and cellulose. And very astonishing fact is that all these three polysaccharides are built from one monomer, one type of monomer. This is glucose, of course, but their chemical and physical properties, I mean this polysaccharide, are very different due to the distinct ways that the monomers are linked together. Yes, and you see it clearly on the picture how these glucose molecules are connected to each other in case of glycogen, in case of starch, and in case of cellulose. And please take into account that starch and cellulose belong to plant polysaccharides, while glycogen belong to animal polysaccharides because, for example, our cells accumulate glycogen and not starch, of course. Another group of organic substances and macromolecules are lipids. So lipids of importance in cellular function include fats, steroids, and phospholipids. So we're going to discuss briefly these three representatives of lipids. First are fats, of course. They consist of glycerol molecule linked by ester bonds to three fatty acids, and we already discussed that fatty acids are essential components of fats. So the eventual molecule of fat is termed as a triacylglycerol or triglycerides because really we have three fatty acids. You see it on the picture, three fatty acids that are hydrophobic part because they can't dissolve in water. And the head part of this fat that contains glycerol that can be dissolved in water. That's why this head part we call we characterize as a hydrophilic part, and the tail part that contains fatty acids we characterize as a hydrophobic part of fats. What can we say? This kind of molecule that contains both, hydrophobic and hydrophilic regions are said to be amphipathic. And also we can say the same word for phospholipids, yes? Fatty acids can be saturated and unsaturated. For example, it depends about the presence or absence of double bonds inside this fatty acid. These representatives that lack double bonds, such as stearic acid, for example, are described as saturated, and some fatty acids that possess in their chemical structure double bonds are unsaturated. This is the fat that we already described here, and also we must describe another group of lipids, very important lipids, because phospholipids are major components of plasma membrane. That's why we must know some information about phospholipids. Their molecules, chemical structure of phospholipids are very similar to fat molecules, so-called triacylglycerol. Only difference is that They contain only two fatty acid chains rather than three. That's why we call them deacylglycerols. Besides this, we have phosphate group inside all kind of phospholipids. That's why we call them phospholipids, because they contain phosphate group, and also one additional polar group such as choline, for example. Yes, and we can describe the structure of phospholipid using this picture depicting these two fatty acids in the structure. That is hydrophobic region of phospholipid, of course, because fatty acids don't dissolve in water. And as for head part of phospholipid, it contains glycerol again, that is connected with phosphate group and connected to choline in this case, because this phospholipid belongs to phosphatidylcholine molecule. But also we can have phosphatidylserine, for example, phosphatidylethanolamine, and other types of phospholipids that are major parts, as I told you, of plasma membrane. And the last group that we are going to describe from lipids, they are steroids. Very important representative of steroids is the cholesterol molecule, a component of animal cell membranes, and also as a precursor for the synthesis of number of steroid hormones such as testosterone, progesterone, and estrogen. So please remember that cholesterol plays many several roles inside the cell, but also we must remember that cholesterol is largely absent in plant cells, so we can't find any cholesterol molecules in case of plant organisms, but scientists believe that plant cells may contain large quantities of related compounds, I mean similar compounds, but not cholesterol, of course. The last group of macromolecules that we must discuss are nucleic acids. Please remember that building blocks for nucleic acids are nucleotides, and every type of nucleotide consists of three distinct parts. Here we have a sugar, we have a nitrogenous base, and a phosphate. These are essential parts of nucleotides as a monomer for nucleic acid. According to nitrogenous bases, we have some differences, of course. If a nucleotide contains guanine, for example, as a nitrogenous base, we say that this nucleotide is a guanine. Yes, and these nitrogenous bases are distributed in two different groups, and the main difference is their chemical structure, of course. The nitrogenous bases that contain two-ring structure, like guanine and adenine, they belong so-called purines, but the nitrogenous bases that contain only one ring in their chemical structure belong to pyrimidines. Yes, and so please remember that cytosine, thymine, and uracil belong to pyrimidines, and guanine and adenine belong to purines. Also, we have differences regarding DNA and RNA molecules. For example, DNA, we must know, contains 4 nucleotides. Among them is thymine, but in case of RNA molecule, instead of thymine, we always have uracil, and this is characteristic to RNA molecules. Sugar part, as for sugar parts of the nucleotides, we can meet here two pentoses, deoxyribose and ribose. And please remember that deoxyribose always is present in DNA nucleotides, and ribose is, as a sugar, is characteristic to RNA nucleotides. Yes. So I want to tell you, for example, this part, because we already discussed that every nucleotide contains 3 parts. Yes, nitrogenous base, sugar, and phosphate. And if we take RNA nucleotide, for example, if this adenine as a nucleotide belongs to RNA nucleotide, it must contain ribose as a sugar and not deoxyribose. You already know, I think, that in every type of cell we can meet two types of nucleic acids, DNA and RNA, or deoxyribonucleic acid and ribonucleic acid. Briefly, very briefly, how these nucleotides are connected to each other. As you can see, according to the picture, one nucleotide with its three components is attached to another nucleotide with the help of sugars and phosphate groups. And please remember that the one sugar from the first nucleotide is attached to the another sugar of the another nucleotide with the help of phosphate group. And that's why we call this kind of covalent bonds phosphodiester bonds, because actually we have two bonds here. One bond with this first sugar and phosphate, and the second bond between this phosphate group and second sugar molecule. And also you can see here clearly formation of these bonds in case of DNA strand or in case of RNA strand. This is very good table depicting the differences between DNA and RNA molecules, and please carefully read it. Somehow we can summarize these differences with the help of this table. For example, if we take function of these molecules, we can see that DNA the main function of DNA is to store hereditary information or genetic information, while the main function of RNA is to read and realize this hereditary information. Briefly about RNAs, they are very also complex structures. They contain one strand, of course, but this one strand of RNA can form some secondary and tertiary structures, and even some RNAs could act like a catalyst. I mean that they can carry out some enzymatic reactions. This is quite new information, and we call this type of RNAs ribozymes. Yes, and we will discuss some examples of it when we go to a transcription and especially translation part. And very important representative of nucleotides, this is ATP molecules, so-called adenosine triphosphate. They are very important molecules because most of the energy in living organisms are derived from this nucleotide. It belongs to RNA nucleotide because it contains ribose as a sugar. It contains adenine as a nitrogenous basis. It contains ribose as a sugar, and 3-phosphate because it contains really 3 phosphate groups. And also, together with ATP molecules, sometimes cell uses guanosine triphosphates, another nucleotide, also of big importance in cellular activity. So please remember that these molecules belong also to nucleotides. So today we summarized and we discussed 3 types of macromolecules, and on the next lecture we will talk about the last group of macromolecules that are proteins, of course. Yes. Thank you very much for your attention, of course.